semester, what I said I would do in the syllabus is that I'm going to give you a sheet with my tests are pretty much 50% multiple choice, 50% short answer, graphing, drawing diagrams, things like that. And what I said I would do is give you a sheet with numerous possible questions. I haven't written it up yet. I've got to get this to you by Thursday. Numerous possible questions for that short answer section. Obviously, I'm not going to give you sample questions on the multiple choice, but I am going to give you sample questions for the short answer and the graphing and the diagrams. And then I'm going to choose from that, randomly in theory, choose from that pool of questions and put that on your exam. So in effect, you have a study guide. So I want to get that to you by Thursday, about 48 hours from now. And then you'll have an entire week to have that, to study it and work on it. What I typically do in my classes is the day before an exam, I always try to hold a review session. So again, that's going to be a little bit experimental. Because I'm giving you questions, don't really want to stand up here and have you guys just say, tell me the answer to number 5, tell me the answer to number 7. That's tedious and it's not conducive to the kind of learning I want you guys to have. So what we're going to do instead is we're still going to have a review session, but I'm going to break you up into groups and you're going to teach each other. So I'm going to be here, I'm going to facilitate this, I'm going to be raising hands that pop up at any time. But then we'll just break up into small groups. Some of you are naturally with your friends. Cool, whatever you want. You want to get into a different group, whatever makes you happy. I'll break you up into groups of, you break yourselves up into groups of 2 to 6, and you go through those while I'm here with you to help you. And it'll be a review session in the sense that I'm here to guide you and make sure you're getting all the concepts clearly. But also, it's going to benefit you to a greater extent because you're going to be teaching it to your friends. And there's no better way to learn except you're going to teach it to somebody else. So that's the way that's going to go. Let me also quickly give you some pointers on my tests. As I said, they're about 50% multiple choice in terms of points and about 50% short to medium long answers, short to medium answers. The multiple choice can get a little tricky. I am definitely known to do that thing where I'll say A, B, and C, and then A, B only, A and C only, etc., and then B above, that kind of thing. And those of you that have had them before are aware. The short answers, I definitely like graphing, and I'm fairly true to my word if I tell you I really like the slide, like the standard growth curve for bacteria. It's pretty amazing that didn't end up on the test. So you can trust your notes. So with that, don't read the textbook cover to cover. Study from the notes. Those of you that are recording, great. Those of you that take good notes, fantastic. Study from your notes. And then use the supplement, use the textbook as a supplement. So your textbook goes into far greater detail, far more complexity, far more material than I ever go into in this class. Not because I don't think it's valuable, but just because I want to keep the scope of a course at a certain level. And this textbook goes a lot deeper than that. So if you're sitting there before every lecture, before the exam, trying to read through the textbook cover to cover, you're drowning yourself, you're overwhelming yourself. And you're probably pretty stressed by that. Don't do that. Go through the notes. Yeah.
So basically, like read the parts of the textbook that we covered in class.
Thank you for saying that. He said basically read the parts of the textbook that we covered in class. Absolutely. Almost every image I put up on a slide came out of your text. Almost every one of them. So you go back, and especially if it's a concept you're a little rough on, you go back and you read the textbook as it related to that image, or that concept that I brought up. If I didn't bring it up in class, it's not going to happen. I write the test as I flip through my notes. Okay. All right, more questions? Okay. Okay, any questions about the review session or anything like that? You guys are in good shape. Cool. All right, so let me quickly, before I get into metabolism today.
Is that a drill?
Before I get into metabolism today, I got an email with some good questions, and let me go over these concepts. You guys frequently ask really good questions. You don't ask them in class, and then your peers don't get the benefit. So I encourage you to ask your questions in class so that everybody benefits from your thoughts. So this student asked me, what is the purpose of a cell envelope? So that one I would like you guys to answer. Someone in the audience, tell us, what is the purpose to a bacterial cell, or you could even scale up in some senses analogous with a plant cell wall. What is the purpose of an envelope in a bacterial cell? There's a lot of ways to get this right.
Improve the efficiency of all the actions inside the cell, because everything will be in tight spot.
Everything in tight spot, hold it together. Structure, absolutely. What else? What are the components of the envelope? Something you want to know about Thursday? What are the components of the envelope? Yes.
Peptoglycogen.
Peptidoglycan, good. What else? There's a murmur, you said the same thing, there's some other murmur back here. Is the peptidoglycan, which is the cell wall in bacteria. What's on the outside of that cell wall in gram-negative and on the inside of that cell wall in gram-positive? Membranes, etc. And what's important about a membrane in a gram-negative cell? Periplasmic space is big. There's a lot of activity going on there. There's a lot of structures on the outside of the gram-negative membrane. Remember those structures? Okay. LPS, polysaccharide. Definitely something you want to know for the next week of your life. LPS is something that's associated with pathogenesis and invasion of the host immune response. So the envelope is primarily contained of the cell wall and those membranes. And so it functions to give the cell shape, to protect it from osmotic stress. Okay? So remember, if you put that cell into a hypotonic media, the water is going to flood into the cell, and the cell wall is going to be rigid enough it's going to help keep that cell from exploding. It protects it under osmotic stress, and it also has a lot of functions in terms of, certainly with respect to gram-negatives, pathogenicity, also adherence, allowing them to stick to things. And I alluded in a couple of lectures ago that bacterial cells really want to stick to things. In many environments, they're going to benefit by sticking to things. All right, cell envelope. Okay, the other question was, let's talk about, we need to step you through again through the endosymbiotic theory. So who here remembers what the endosymbiotic theory is?
Isn't that when one bacteria grew inside another bacteria, and then therefore it became the eukaryotic cell eventually?
Yeah. Okay, great. He said one bacteria grew inside another bacteria and it eventually became a eukaryotic cell. Good. What else? Can you give a little more detail?
Some heterotrophic bacteria become mitochondria, and autotrophic bacteria become chloroplasts.
Excellent, excellent. So a heterotrophic bacteria, if another cell engulfed another heterotrophic cell, that might have eventually become a mitochondria. Okay? And if that cell engulfed, say, a photosynthetic cell, that would have ended up being the precursors of a chloroplast for our eukaryotic cells. Eukaryotic plant cells versus non-autotrophic cells. Yeah?
Question. This thing that a cell can ingest all material and go through the endosome and goes to the lysosome, this is also part of the endosymbiotic theory?
No. Good question. So you're just talking, so here, let me tell you why I know it and why I think you think that. So he says is just the act of endosymbiosis that we talked about for eukaryotic cells, he said, is that part of the endosymbiotic theory? And I said, no. But there's a reason he thinks that. And the reason it's not part of it is that's just the way on a day in, day out basis, a eukaryotic cell can ingest everything. Pinocytosis, in solvent, or in solution, or formal endocytosis, where they'll actually wrap the membrane around and take in a particle. And form a lysosome to digest that particle and actually consume that particle. Think about a classic amoeba just making pseudopods and wrapping its arm around something and taking it in. But there's a good reason he thinks that. There's a good reason he thinks that's part of the endosymbiotic theory, and I would say however many billions of years ago that we first saw the first eukaryotic cells emerging, it very well may have gone something like that, where it was an missing my word, not endosymbiotic, endocytosis event, if that's a word, the act of consuming a cell, and it became somewhat parasitic in it. It didn't digest it, it became parasitic. Here's an analogy that's going on right now that you see. You have a species of Paramecium. Forget which one. Maybe Bursaria, but don't quote me on that. You You have a species of paramecium. That paramecium can harbor symbiotic algae or not. So you'll see examples of this paramecium swimming around. It's a paramecium, which in every textbook would say is a heterotroph, okay? Right? Not an autotroph, a heterotroph. You'll see this paramecium harboring symbiotic algae with it. The symbiotic algae benefit because they get a habitat and they get this nice nutrient source from living on the inside of this paramecium. Paramecium benefits because it's got this nice plant on the inside capturing all that carbon, right? But that paramecium can live perfectly happily without that symbiotic algae as well. So it's not gone as far as to be a permanent symbiosis and the permanent state of being a functioning primary producer plant cell that has a chloroplast, but it does harbor algae. Very likely takes it in that way, but I'm not sure. Probably also passes it from generation to generation. Okay, those are good questions. Anything else? Okay. All right, so let's get into metabolism. I don't want to give a short service to metabolism. All right, so for many of you, aspects of this will definitely be a review. We're going to go through things, just a very superficial looking at enzyme chemistry, and then we'll start to get into actual metabolism. At the end of the lecture, we'll get into glycolysis and the Krebs cycle just a little bit. And I'll help you out. I'll kind of tell you what you would need to know for a test and not for a test. All right, so metabolism is really the sum of two things. Okay? So it's the total of all chemical reactions in a cell, and it's the sum of two things, metabolism and anabolism. Who here can tell me the difference in metabolism versus anabolism?
Metabolism is breaking down, anabolism is synthesis.
Excellent. Were you going to say something?
I was gonna say catabolism, it synthesizes the energy, where in anabolism, it more creates energy.
Good, well put. So he says, and you're both absolutely correct, excellent. So in metabolism, you're breaking things down, and in the process of breaking things down, big molecules, glucose, proteins, fats, etc., you release a lot of energy. Energy from catabolic reactions, energy that gets released in catabolic reactions, gets stored in what molecule? Anyone know? Not everybody. No. Glucose is getting broken down to store this energy in
Glycogen?
later on, too far down the road. ATP. But good, you're not wrong. All right, that energy gets stored in ATP, and then later on, that ATP gets spent like currency, so that that cell can build new molecules. It can build nucleic acids, it can build amino acids, proteins, etc. And maybe build glycogen. All right. All right. So, if you remember from the last lecture last week, we talked a lot about nutrition, and I talked about the basic fundamental needs. We all need nitrogen, carbon, phosphorus, etc. You know that, we're pretty comfortable with that. But we also need three basic things in three forms. So, every organism needs some kind of an energy source, some kind of a carbon source, and some kind of an electron source. All right? And the analogy I gave you guys is us. We're class of heterotrophs. We get our energy, our carbon, and our electrons all from one source. And that's reduced carbon compounds, proteins, sugars, fats, etc. All right? Not all microbes do that. All right? So let's break this down. The energy sources, you can have a phototroph, getting its energy from light. A lot of autotrophs do that. Cyanobacteria, algae, higher plants, etc. And then you can also have a chemoorganotroph and a chemolithotroph. All right? And the chemo is referring to the fact that it is not getting its energy from light, it's getting its energy from chemicals. Those chemicals can be organic, organotroph, like us, or they can be inorganic, is a chemolithotroph, like an organism that uses hydrogen sulfide,
Listen.
hydrogen sulfide, ammonia, etc. for its energy source. So you'll notice the organotrope part and the lithotrope part here mesh with the electron source. Those things overlap. You're not confused. Yes, they do overlap. Don't try to separate them in your mind because we have three little boxes here. They really do overlap. So if you're thinking, well, it's the same thing, it is. You're on the right track. So in this case, coming up here with the chemoorganotrope, in this case, they're getting it from organic molecules. A little bit repetitive here. And the lithotrope is from inorganic molecules. We all need carbon from somewhere. Heterotropes, organic molecules, sugars, proteins, etc. And then autotrophs fix inorganic carbon dioxide out of the atmosphere and turn it into those sugars. So autotrophy, we're really going to talk about more on Thursday, because that's, if we're talking about catabolism and anabolism, where is autotrophy going to fall?
Anabolism.
Anabolism, good, okay? Whereas heterotrophy, we're taking in those sugars, we eat that protein, and we're breaking it up, catabolism. And we're scavenging those electrons, etc. Oh, okay, so that was the review part, sorry. What we're all trying to do is just build new cells. So, the energy source is what fuels the generation of ATP. Alright, so breaking down all of those reduced molecules, whether it's inorganic like hydrogen sulfide or whether it's organic like glucose. Breaking that down, breaking apart those molecules fuels the generation of ATP. We're going to go into a lot of detail on that later in this lecture. The carbon source, the breakdown of those things, the breakdown of glucose, is what makes precursor metabolites. Alright? So here's your anabolism. Those precursor metabolites that were broken up here use the energy that was stored in the ATP to form other building blocks. And you need electrons feeding them and make bigger and bigger and bigger, more complex things. Okay? Like those building blocks is ultimately macromolecules and structures like cells. So this is basically the catabolism, breaking up, using the energy, using the electrons to do anabolism. Okay. Quick reminder, catabolism is what generates energy. Provides a ready source of reducing power, those electrons that you're going to need later on for biosynthesis. And it generates the smaller carbon molecules, breaks up big carbon molecules and small carbon molecules, so they're the precursors, the building blocks of later things that cell wants to make. Anabolism is the opposite. It requires the energy from catabolism. Okay? And it's making complex molecules out of simple ones. Okay, so what does ATP do? ATP basically fuels these reactions by giving its energy to it. So ATP is adenosine triphosphate. Remember that? So it's got 3 phosphates hanging off of it. Very high energy bonds. Very high energy bonds, very valuable, storing a lot of energy. So if you have a basic inorganic reaction where you have ATP getting converted to C to D, remember your basic chemistry just based on the arrows, we're showing it's inorganic because the precursors are more energetic than bigger. You have to put energy into this system to get over to C to D. In many cases, this is just an analogy, but in many cases, the way that energy gets put in is through the hydrolysis of ATP to ADP plus inorganic phosphate. So by cleaving off, if you couple the reaction, when you cleave off that inorganic phosphate, you release that energy, couple it to the reaction, such that moving to C to D becomes favorable. You expend energy to go what would otherwise be chemically uphill. All right, everyone comfortable with that? Okay. So let's talk about redox. Raise your hand if you're comfortable with the idea of redox. Okay. No problem. You don't have to be that comfortable. Okay. So redox is just reduction and oxidation. All right. So go back to your chemistry. What is the reduction of a compound? If I'm reducing nitrite, where's it going to go? How does that? You tell me. What are you going to say?
You're going to add an electron to the nitrate.
Exactly. You're going to add an electron. Exactly. To nitrite.
Nitrate.
To make it nitrate. Right? Yeah. Okay. So it's the adding of electrons. All right. So if you have that base nitrogen molecule, all right. So if you add an electron to nitrite, that basic nitrogen molecule, no, I'm wrong. I am wrong. There you go. Better? All right. Adding electrons to a molecule is reducing it. Removing electrons from that molecule is oxidizing. All right. Adding electrons to a molecule expends energy. You're putting more energy into that molecule by having more electrons on it. All right. So the more energy rich, the more electrons the molecule has on it, the more energy rich it is. Okay. And you can use that energy. So later on, we're going to do just basic NAD plus.
Nah.
All right. So we're going to reduce nicotinamide to NADH. And that's going to be a storage molecule for energy and electrons. You'll see that when we get into the electron transport chain. All right. So this movement of electrons back and forth is basically an energy exchange. And there's a redox potential. They're not all created equal. Okay? An oxygen molecule, obviously, is highly oxidized. Sounds. All right. It doesn't have a lot of electrons on it, and so it very readily accepts electrons. Okay, so where is oxygen in the electron transport chain? Very, very end. Good. And it's at the very, very end because it's energetically most favorable to accept electrons. All right? So the more negative that E sub O is, the better it is an electron donor. So think about it. Electrons have a negative charge. The more negative that E sub O is, the more electrons it has to give up. All right? The more positive E sub O is, the better it is as an electron acceptor. Oxygen is an extremely good electron acceptor. Glucose, a highly reduced carbon compound, is a very good electron donor. Hydrogen sulfide, very good electron donor. That's why organisms use those things for energy. That's why we eat glucose. That's why bacteria want to eat glucose. That's why bacteria in some cases want to eat hydrogen sulfide or ammonia. And so let's look at that on the scale. So this is a scale of reductive potential. Okay? That E sub O. So the compounds that have extremely negative E sub O, things like glucose. There's a reason we want glucose. When you go back to your cultures in a lab, you feed glucose. It's very energetically rich. Really easy to use and get a lot of energy out of it. All right? The most positive down at the bottom is oxygen. Oxygen is your best electron acceptor. So the difference, the biggest difference you can move across this scale is going to be the most energetically favorable, the most downhill energetically. All right, so I just said we're going to go from NAD plus to NADH. All right, so then NADH, excuse me, is a very good electron donor. Nitrite is a pretty good electron acceptor. Oxygen is the best. All right? So there's a scale along there. And where does NAD come into the electron transport chain? Don't remember? You don't necessarily know this yet. I haven't taught it to you yet. Does anyone remember from other classes? All right, we'll get to that.
Before going to the side,
Not quite. It's out of the Krebs cycle. It's out of the Krebs cycle. We'll get to that by the end. Just don't sweat it. All right, and you can look at some of these half reactions, all right, in terms of their energy. So obviously, the reduction of oxygen is very positive because it really wants to accept those electrons, and you can look at some other things. So going back to the electron transport chain that we will get to certainly by Thursday and a little bit at the end of this lecture. The cytochromes, jarring your memory a little bit. Remember, cytochromes are the molecules that are along the electron transport chain that transfer the electrons down. We really want to transfer those electrons downhill in terms of their affinity. Okay? Whereas the highest affinity is oxygen. That's why it's at the very end. And then things like the cytochromes, cytochrome B, cytochrome C, slightly decreasing affinity. All right, okay. Are you guys comfortable with this before I move on? I'm seeing either people sleeping or puzzled faces. You might even be shopping for shoes. Anyone? Anyone? Okay. All right, so let's jump over to enzymes. So what enzymes do is they lower the energy required to move these reactions forward. All right? They carry out reactions in physiological conditions so that they can happen faster. So something that would otherwise happen kind of slowly because it's not very energetically favorable. Enzyme can facilitate that and speed that up. Speaking of the electron transport chain, when we get to ATP synthase, a very famous enzyme that spans the membrane, that's a classic case where the hydrogen ions would normally just slip back across the membrane. ATP synthase facilitates that, makes it go much, much faster, and generates ATP. So they speed the rate of reaction, and they lower the activation energy. And so the way an enzyme works is just basically, here you have, this is just kind of a silly cartoon. You have an enzyme, these are the substrates. So this is A and B, basically, A plus B. The enzyme takes it, alters it in some way, so then you end up with this complex where you have the substrates and the enzyme together. Right? And then the enzyme does whatever it's going to do. This is obviously really generic. And spits out a product, CE. Okay? So you have this complex working together. And so as I was saying, it lowers the activation energy. It's really easy to look at this. The best way to look at this, is graphically. So here you have progress of a reaction. Okay, you have A to B trying to turn into C to D. All right, and this is a reaction that would normally take place very slowly, but the free energy required to make this reaction take place is pretty high. So follow the purple line. So if I want to convert A to B to C to D, if I want to convert A to B to C to D, I have to go through very, very high activation energy in that transition state complex. It's a very high free energy. What the enzyme does, though, is it basically just reduces that transition state such that it requires less energy. So the hump it has to go over to convert A to B to C to D, the hill it has to go over, becomes much, much slower because of the ability of an enzyme to facilitate that reaction. want you to understand, even if you don't necessarily know. So the idea is that enzymes, they function, they facilitate the movement of a reaction. Great analogy is ATP synthase. It's like a revolving door. And they facilitate this reaction and they make it move faster. So you basically have this little machine that's taking A and B and converting it to something else and spitting out C and D. But that machine can get saturated and it can only move so fast. Okay? At some point, if it's fully saturated, it can't move any faster. Okay? And so you have, as the substrate concentration, as the AB components get more and more and more concentrated, in theory, that enzyme is going to work harder and harder and harder. Right? It's going to work harder and harder and harder, and go faster and faster and faster. This is the velocity of the reaction, or the enzyme speed reaction. The velocity of that reaction is going up, it's going faster and faster and faster, until some point at which it's so saturated it can't move any faster. It's at its maximum capacity. It's at its Vmax. The rate of reaction is at its fastest.
Could you also say that, or it makes sense to say that free energy is available to keep up at that point?
Think about your graph. That's a really good question. He's saying, well, could you also say the reactant is used up? Look at your x-axis. So if the reactant is used up, we will start slowing down. So if we're having less and less and less A and B, then actually it's going to start slowing down here in terms of velocity. This is a rate on the y-axis.
Okay.
Okay?
You said free energy, not reactant. Wait, free, you said free energy, right?
Yeah.
Oh, I thought you said the reactants. Oh, man, totally. How did I not hear that at all? Please ask it again. I apologize.
As the substrate concentration increases and the enzyme has to work harder, does that mean the free energy is decreasing?
Oh. 100 no. The free energy is associated with just the step of when an individual reaction is converting it. That chemical energy is converting A to B, C to D. Okay? And it's related to this individual. Okay? No, I don't necessarily tie this together. I don't tie this. I see this as an individual level. A to B requires this much energy to get to C to D.
Okay.
Design makes it use a little less energy, a little less energy. Okay? Whereas this is a really good question for clarifying what I'm saying. Because what this is looking at enzyme chemistry in a soup. You have a mixture here, and you have so much enzyme, and as the substrate concentration gets more and more and more, you're going to maximize the output of that enzyme. So whether it's just one enzyme in the solution, one molecule of enzyme, or a million molecules.